Is one glass of wine really safe? A deeper scientific look. For more than 40 years, we've been told something comforting, that moderate drinking is not only safe, but potentially beneficial. A glass of wine for women, 2 drinks for men. Some even suggested that abstaining completely might increase cardiovascular risk. It sounded balanced. It sounded scientific. It sounded responsible. But when we look at the data more closely, especially more recent, better controlled analyses, that conclusion becomes much less convincing. This is not about moral judgment. It is not about prohibition. It is about biology. Let's examine what alcohol actually does inside the human body. Where the healthy alcohol narrative came from. Much of the belief in moderate drinking came from observational epidemiology. Researchers observed a so-called J-shaped mortality curve. Heavy drinkers had high mortality, non-drinkers had moderate mortality, and moderate drinkers appeared to have the lowest risk. On the surface, that looked protective. But there was a methodological flaw. Many people in the non-drinker category were former drinkers, individuals who had quit because of health problems. When lifetime abstainers were separated from former drinkers, the protective effect largely diminished or disappeared. This is called confounding bias. Association is not causation, and when large data sets were reanalyzed controlling for these variables, the idea that moderate drinking protects the heart became far less certain. More recent global analyses have suggested that the lowest health risk is associated with very low or zero intake. Alcohol is not treated as a nutrient. Your body does not metabolize alcohol the way it metabolizes carbohydrates, fats, or proteins. It treats alcohol as a toxin. The moment ethanol enters the bloodstream, the liver shifts its priorities. Detoxification becomes primary. Other metabolic processes are temporarily deprioritized. Ethanol is converted by the enzyme alcohol dehydrogenase into acetaldehyde. Acetaldehyde is highly reactive and toxic. It forms adducts with proteins and DNA, disrupts cellular structures, and increases oxidative stress. Acetaldehyde is then converted into acetate by aldehyde dehydrogenase. But the intermediate steps matter. During alcohol metabolism, the NAD+ to NADH ratio shifts significantly. This redox imbalance affects multiple metabolic pathways, particularly those involved in fat oxidation. When NADH accumulates, fatty acid oxidation slows, triglyceride synthesis increases. Over time, this contributes to fat accumulation in the liver, even in individuals who are not heavy drinkers. Non-alcoholic fatty liver disease and alcohol-associated fatty liver share overlapping metabolic mechanisms, and fatty liver is not harmless. It is strongly associated with insulin resistance, systemic inflammation, and cardiovascular disease. Oxidative stress and mitochondrial damage. Alcohol metabolism increases the production of reactive oxygen species. Free radicals are natural byproducts of cellular metabolism, but excessive levels overwhelm antioxidant defenses. The liver is particularly vulnerable, but so are the heart and brain. Mitochondria, the energy-producing organelles inside cells, are highly sensitive to oxidative damage. When mitochondria are impaired, ATP production decreases, inflammatory signaling increases, cellular repair slows. This is not necessarily dramatic damage after one drink. The effect is cumulative. Small exposures, repeated over years, can contribute to measurable changes in tissue structure and metabolic function. The brain, suppression versus resilience. Alcohol enhances the activity of GABA, the primary inhibitory neurotransmitter. That is why people feel relaxed. Anxiety decreases temporarily. But alcohol simultaneously suppresses glutarate, alters dopamine signaling, and interferes with synaptic plasticity. What feels like relief is neurological suppression, not restoration. Neuroimaging studies have shown associations between moderate alcohol consumption and reductions in gray matter volume in specific brain regions, including areas involved in memory and executive function. The brain adapts to repeated exposure, tolerance develops, neural balance shifts, and in some individuals, mood dysregulation follows. Alcohol is often used to cope with stress. Biologically, however, it may increase vulnerability over time. Cardiovascular effects. Alcohol influences the cardiovascular system in multiple ways. Even moderate intake can raise blood pressure. It impairs endothelial function, the delicate inner lining of blood vessels that regulates vascular tone, clotting, and inflammatory signaling. Repeated exposure can promote endothelial dysfunction, a precursor to atherosclerosis. Alcohol also increases the risk of arrhythmias, particularly atrial fibrillation. This effect has been observed even in individuals who consume relatively small amounts. Red wine contains polyphenols, such as resveratrol. These compounds have antioxidant properties in isolation, but they do not negate the metabolic consequences of ethanol. Antioxidants and alcohol coexist in wine. They do not cancel each other out. Alcohol and glucose regulation. Alcohol affects blood sugar regulation. It suppresses gluconeogenesis in the liver, which can lead to hypoglycemia, particularly in fasting states or in individuals using glucose-lowering medications. Later, reactive hyperglycemia may occur due to stress hormones and altered insulin sensitivity. Over time, alcohol can contribute to insulin resistance, especially when combined with poor sleep, chronic stress, and high sugar diets. The metabolic picture becomes complex. Alcohol is not just a drink, it is a systemic metabolic event. Micronutrient depletion. Alcohol interferes with the absorption and utilization of several key micronutrients. Vitamin B1, thiamine, is particularly important. It plays a central role in carbohydrate metabolism and neuronal function. Deficiency can impair nerve signaling and energy production. Magnesium levels may decline. Magnesium is essential for hundreds of enzymatic reactions, including those regulating muscle relaxation and nervous system stability. Zinc and potassium can also be depleted. These minerals influence immune function, cardiac rhythm, and fluid balance. Many subtle symptoms, fatigue, irritability, mild neuropathy, sleep disturbance, can be influenced by nutrient status. Alcohol and sleep. Although alcohol may help some individuals fall asleep faster, it disrupts sleep architecture. REM sleep is reduced. Sleep becomes fragmented. Deep restorative phases may be shortened. Sleep is a primary mechanism for neural repair and metabolic regulation. When sleep is impaired, cortisol increases, blood sugar rises, and inflammatory markers may elevate. Thus, alcohol and stress can form a reinforcing cycle. Drink to relax, sleep worsens, stress increases, drink again. Can the damage be reduced? Nothing neutralizes alcohol completely. However, metabolic resilience matters. Hydration and electrolyte balance support cellular function. Thiamine and magnesium repletion may mitigate some effects of deficiency. A diet rich in antioxidant-containing vegetables supports endogenous defense systems. Regular physical activity stimulates mitochondrial biogenesis, the creation of new mitochondria. This improves metabolic flexibility and stress tolerance. Intermittent fasting may enhance autophagy and cellular repair processes, though this should be individualized. These strategies do not erase alcohol's effects, but they improve the body's ability to recover. Alcohol in the context of overall lifestyle. Risk is cumulative. A single glass of wine on a balanced, nutrient-dense, physically active lifestyle may carry different consequences than alcohol combined with poor sleep, chronic stress, sedentary behavior, and high sugar intake. Health is rarely about a single variable. It is about patterns.
But it is important to separate cultural habits from biological realities. Alcohol is widely normalized, it is socially embedded, but from a purely physiological standpoint, it is not a health-promoting compound. Final perspective. Moderate drinking is not a medical intervention. It is not a cardiovascular therapy. It is not a metabolic enhancer. If you choose to drink, do so consciously. Understand what is happening inside your body. Support recovery. Prioritize sleep. Maintain nutrient sufficiency. Exercise regularly. Manage stress. Health is not about perfection. It is about awareness. And awareness begins with understanding biology, not marketing, not tradition, not assumptions. One glass of wine may feel harmless, but safety is not defined by feeling. It is defined by physiology.